Okay, so good morning, everybody, and welcome to this month's Genetic Code Expansion webinar. We thank those who have joined us previously for coming again and are excited to have any new attendees with us today. We hope you've been enjoying these series hosted by myself and John Lewick and organized by the GCE for All Center here at Oregon State University. We've been humbled and grateful for having so many hosts, or having hosted so many great scientists in the past, ranging from early career graduate students to established world-renowned scientists with expertise not just in genetic code expansion, but also translation biology, tRNA therapeutics, and directed evolution. If you have any suggestions for speakers or are interested in speaking yourself, do reach out to us at the center. Also, we'd like to announce that we're hosting another GCE workshop this summer, July 28th to August 1st, where we will be training attendees in genetic code expansion technologies for installing post-translational modifications. Please spread the word, and we'd be excited to host you here in beautiful Corvallis, Oregon. Hey, today we are excited to introduce two speakers, Drs. Amanda Jarvis and Tao Liu. First up is Dr. Amanda Jarvis, currently Senior Lecturer and Independent Scientist at the University of Edinburgh. Amanda graduated from the University of St. Andrews in 2007 with a master's in chemistry, during which she spent a year working in the petrochemicals industry in Calgary, Alberta. She went on to receive a PhD from the University of York under the supervision of Professor Ian Fairlam. Amanda joined the group of Dr. Philippe Daubin as a postdoctoral research fellow in 2011 and worked on the development of ruthenium-catalyzed nitrene reactions. In 2013, she moved to Professor Paul Kamer's group to work on the development of artificial metalloenzymes. And in 2015, she received a Marie Curie Individual Fellowship to continue working in Professor Paul Kamer's group on artificial metalloenzymes for the oxidation of alkanes. Amanda started her independent career at the University of Edinburgh in the fall of 2017 as a Christina Miller Fellow and was awarded a UKRI Future Leaders Fellowship in May of 2019. In 2022, she was promoted to Senior Lecturer. Her research seeks to design transition metal catalysts inspired by the architecture of natural enzymes that are selected for unnatural reactions such as carbon-hydrogen amination. Ultimately, her goal is to design environmentally sustainable catalytic chemistries, including those that can be achieved using GCE to encode metal binding ligands such as pyridyl-triazines, which she will talk more about today. Thank you for joining us today, Dr. Jarvis. Please take it away.
Thank you very much. Let me just share my screen. That's all showing correctly?
It looks good.
Great. So thank you very much for the invitation. It's great to be one of the presenters here. I've seen a couple of the webinars that have gone on. And as you may have kind of noticed in that introduction, my background has been, or originally was an organic chemist, an organometallic chemist, looking at methodology. And then slowly over time, I've moved towards the more biological. And today I'm going to tell you a couple of stories about the work we've been doing with non-canonical amino acids. So generally what motivates me and my research group is whether or not we can design completely new to nature active sites in enzymes. And from my background in kind of transition metal chemistry, we're really interested in whether we can design new metalloenzymes and introduce very different coordination spheres to what nature would have. And our interest in doing this is to see if we can actually introduce some of the sort of classical industrial chemical reactions into the biocatalytic toolbox, which might help us to kind of end up with greener synthesis going forwards and kind of helping the planet reach some of its kind of targets and things like net zero. But at the same time, we're also sort of aware that with these different active sites and different protein architectures, we may also end up creating systems that could have solutions or applications in a number of different areas, including potentially health and materials. So where I started in this field was kind of in the sort of area of artificial metalloenzymes or sort of biohybrid catalysts, where we were interested in taking different protein scaffolds. And so I've shown just two of the protein scaffolds we would commonly use, so SCP and myoglobin. And through using different kind of ligands or metal cofactors, we could introduce a range of different metals. So for example, with myoglobin, you can think about changing your protoporphyrin metal complexes or using sort of similar planar aromatic ligands and complexes. And with SCP, we started looking at whether we could use reactive amino acids such as cysteine to introduce a whole different range of very abiological kind of ligands, metal binding centers into a protein. So for example, this would even include phosphines. But as time went on, we've also been interested in whether we can move away from that sort of bioorthogonal transformation to directly using unnatural amino acids. And the two stories I'll tell you about today include our work on bipyridylalanine and this triazine pyridyl system. And the ultimate goal, or sort of things we were looking at, is whether using this whole kind of library and different systems, we could start to look at reactions such as hydroformylation or palladium catalyzed cross-coupling, where, just where nature isn't working. And ultimately, whether we could provide enzymes that we could then introduce into biosynthetic pathways, allowing us to start from natural substrates to build very different complex unnatural products. So to start with, I'll tell you how we started in the field of the sort of non-canonical amino acids with bipyridylalanine, and the work that we were also doing with some of our cysteine conjugation using the steroid carrier protein 2L shown here. So to set the scene, why this protein scaffold? Well, we chose this protein because it was a known structure, so we had a crystal structure. It was relatively easy to produce with good yields and easy to modify. So this is the crystal structures shown here, and one of the features you can see it has is this apolar tunnel. And our thought was that in reactions where you might want linear selectivity, could we use this tunnel to help direct selectivity? The protein scaffold is quite small, and there are no, or one of the reasons it was attractive for our first set of work was that there were no cysteines present, so we could introduce the cysteine wherever we wanted. And this meant we could then do modification using alkyl bromides or malamides to get through and introduce our ligand, which could then bind a metal through the 2-nitrogen ligands. And we could get through to these cysteine-modified mutants in relatively high yields. We were also interested in exploring this, and went on to show we could also use this protein in genetic code expansion. So I'm sure for this audience, this is a slide that probably doesn't need explaining, but we're using amber stop codon suppression. And at the time of gene expression, we're then adding our bipyridylalanine, and we're using relatively large amounts of this. So luckily we have a background in organic synthesis, so we're able to make the bipyridylalanine on kind of a 10-gram scale, allowing us to get through to our proteins of interest. And so what we have done with this is we were able to look at a couple of different positions in the protein, Q11, A100C, and B83. So these were the natural positions which we replaced with bipyridylalanine. And if you remember that apolar tunnel, these are situated in the middle of the tunnel and then at either end. So they're kind of chosen to see if we can explore the space around that tunnel. And we use mass spectrometry as kind of our go-to tool to kind of show that we are able to incorporate these amino acids cleanly into our protein of interest. And then in a couple of cases, we've also been lucky enough to get crystal structures, which also show quite nicely bipyridylalanine sitting within the protein structure. So the first thing that we wanted to try was actually a very simple kind of experiment where we wanted to compare how a steroid carrier protein where we'd introduced a cysteine and then bioconjugated a bipyridylalanine would compare with a protein where we'd used the unnatural amino acid bipyridylalanine directly. And our sort of thought process here was when we're doing the orthogonal kind of modifications, we have to go through a couple of separate steps. We end up purifying the protein, whereas with the genetic code expansion, you can maybe more quickly and easily access your protein of interest. But we just wanted to see how, whether that was true, or in our hands, how easy it was, and then also whether or not there was any similarity in what you would get from the results for using either or, because one of the advantages of the cysteine kind of modification route is you can very easily change what cofactor or ligand you're using, whether with genetic code expansion, that might take a lot longer if you're having to evolve your translation system to accept it. And so the first metal we looked at was just copper binding. This had been described a couple of times in the literature. And what we found is if you titrate in copper to both of these proteins, you see the same sort of redshift showing that you are binding the copper between the two nitrogen ligands and getting through to a copper metalloprotein. And we were able to then also confirm this with X-ray crystallography done in collaboration with Julia Richardson at Edinburgh. For one of our positions, so Q111, which is in the middle of the tunnel, we got these two crystal structures, really nicely showing that we would be able to bind our copper to the bipyridine part of either the cysteine modified system or the amino acid. So the next step, as I sort of mentioned at the beginning, our driving interest is really catalysis, and where we can go with catalysis was to see if we could explore this in a catalytic reaction. And the reaction we chose at first was this sort of benchmark Friedel-Crafts alkylation between indole and this enone shown here, which combines the copper at this position. And if you're bound to the copper within the metalloenzyme, then you can expect that you might get some control of the chirality at this position, leading to one or other enantiomer of the product. And what we saw was with our cysteine modified proteins and with the amino acids, we were getting kind of moderate yields, roughly in line with the yields that you might expect just from bipyridine and copper catalysis. But with the cysteine, we were seeing relatively low enantioselectivity, so enantiomeric ratios of only around 65 to 35. But when we kind of use the amino acid, which has a sort of shorter length between the protein backbone and where the copper would sit, we can begin to see that we're actually increasing our enantiomeric ratio up to kind of 20 to 80, which is a relatively good starting point if you want to start thinking about doing further directed evolution to improve this further. The other thing that we sort of noticed that was sort of unexpected is if you take the, if you compare the results from the cysteine modified protein with that of the bipyridylalanine, we seem to switch the preference of the enantioinduction. But if you do look more closely at the crystal structures, this is maybe not so much of a surprise, as you can see, that they do seem to fit in two quite different orientations. And and computational work around this kind of showed that you can model the substrate binding and you kind of mimic the exact ratios that were observed. So we did look at seeing if we could sort of rationally improve our results, particularly in terms to the enantioselectivity. But using both computational methods and the crystal structures, we didn't really identify any positions nearby through aniline scanning or anything that seemed to be having a great deal of difference to the results. And so we think it might be because the position, at least in the Q11 situation, is just a bit too exposed on the surface, and actually further kind of development of the protein coils around these complexes would be needed. So At the same time, we were thinking about what else could we do with bipyridine. So as a chemist, you often see bipyridine in sorts of complexes like this. So your kind of classic photoredox catalysis, or kind of piano stool complexes. Some of these are actually used in as metallodrugs. So those were some of the options we were looking at. And then there was this example, which I'll sort of talk to you through today, where there had been a report in the literature where they'd shown that potentially you could use metal binding to improve bioorthogonal chemistry. So if we just take a step back, non-canonical amino acids for KIT chemistry are very well known. The GCE for All Center has a lot of experience in this. And here I've just shown a range of the different types of amino acids that have been explored, from the sort of azide alkyne coupling partners to your sort of tetrazine ligation systems. The tetrazines in particular have had a large exploration of what they do, and this is due to their exceptionally rapid kinetics, which can be tuned as well depending on what dienophile you use with it. So whether you're using your trans-cyclooctene as shown here, or whether you're using your bicyclononane as shown here, you can get through to very high rates of reaction, which in turn is desirable if you're doing in vivo chemistry or cell labeling and you're wanting to capture events that are happening very quickly. But from the literature, there were some there were some hints that maybe tetrazines, whilst their rates are very fast, this also can potentially lead to other problems, such as being prone to hydrolysis and reactions with endogenous thiols. And so there has been some work kind of 10 years ago from the pressure group that had showed that actually 1,2-triazines were maybe more biologically stable alternatives, but the sort of payoff for this stability ended up being that they had much slower reaction kinetics. And so just before COVID, I was at a conference where I met Valerie Kovsnikov from Northumbria, who is an inorganic chemist who had been working on kind of some small molecules where they've been exploring these triazines, but including a pyridine in to be bound to it, and then using metal binding to look at the rates of the click reaction in this position. And what they were able to show was that if you included iridium, you could improve the rate of the reaction by up to 140 times. And this got you kind of into what was kind of considered the sort of realm that you'd need for in vivo reactions. In addition, they also showed that when you added, when you had your sort of iridium and then added your bicyclononane reagent, you went from something that wasn't emissive to something that was emissive. So there was the potential that you could also follow the success of the click reaction of by looking at the emission of the complex. And so Valerie asked, saw some of the work we were presenting and said, did we think we could look at this as an unnatural amino acid? And so could we consider making an unnatural amino acid that looked a bit like this? So taking the inspiration both from the bipyridylalanine systems we'd previously used and the work of the pressure group. And if so, would we be able to show that they could either bind metals or do a bioorthogonal reaction? And ultimately, if you put the two together, could you actually follow that reaction? So Andre in Valerie's group was able to make some of this triazine amino acid for us, and then he sent it down. And we very simply took the exact same system that we'd used with bipyridylalanine, so the MJ system that had been developed by Schultz, and showed that if we were using that system, it would accept the triazine, and we would get incorporation into our protein of interest. And we once again were using FCP as we were familiar with using it in the past. But in this case, we chose a position on the outside of the protein for our first investigations. And this was just to make sure that if we were looking at the iridium binding later, we would have plenty of space to make those complexes. So the next thing that we looked at, having successfully managed to put the amino acid onto the protein, was would it carry out the bioorthogonal reaction, the sort of click reaction. And so using 10 equivalents of this quite simple bicyclononane, we were able to show that we could get through to complete modification by mass spec of our desired product. And we also looked at a few other examples. So we looked at both a commercial transcyclooctane dye and a pyrene transcyclooctane. And what we could see, at least in this example very clearly, is with the commercial dye, we do see also that we're getting a fluorescent band where our protein is, suggesting we're getting modification of our triazine through with the dye. So that was very nice. We'd shown that we had a new click reagent. So the next step was to look at whether or not it would bind iridium. And so for this, we used a iridium precursor with this phenylpyridine with 2 fluorides. So we used a monomeric iridium precursor. And again, just kind of treatment with these for a few hours led through to a product, which when we looked at the fluorescence gave very different UV or photophysical properties, again indicative that we had bound the iridium to our triazine. So you can see shown here that when we've got our triazine within our iridium, we get a luminescent product with emission at around 600 nanometers. In comparison, if you have an iridium bipyridine small molecule equivalent, so without the triazine and without the protein, you would expect that around 540 nanometers. And if you had bipyridine on a protein, you would then expect that at 490 nanometers. Something to note, this is normalized emission, but we do see a little bit of binding of the protein itself to iridium, although you can tell from the emission that we see very different emission maxima for this product. And visibly, you can also see this. So the triazine is a kind of reddish color, whereas the other systems are bluer in the fluorescence. So finally, we looked at putting this all together. So if we have our iridium, could we still do the click reaction? And what you can see is this is our starting triazine emission. Once we add in the BCN, we move it, it's blue shifted, following this line at 2. And you can see that you do have some overlap with something that's similar to the iridium species. And this is to be expected because you're going to actually change your triazine section now to something that is much more like a bipyridine. And so we felt this was quite good evidence that we were actually able to do the click reaction. In addition, we were wondering to what extent this goes. And so we haven't managed to fully confirm it, but if you look at the emission spectra, once you have the iridium and as well as the BCN, we actually see an increase in the emission. And what's more noticeable about this is actually we lost some of the protein in this experiment. So actually we're seeing we are potentially seeing a little bit of switch on, even though the iridium itself does show emission in our case in the protein system. So it's not exactly like the small molecule work where the iridium complex wasn't emissive. We find with the proteins that it isn't. So we're now kind of thinking about where we go with this project. So that was sort of our fresh initial work. And so it just leaves me to summarize what I've shown you so far. So hopefully you've got a brief idea of how we view metal binding amino acids. So we think they can introduce quite a lot of different functionality. So both as metal complexes for catalysis, but also, as we've shown with this kind of dual functional protein, that maybe we've got options to kind of speed up chemical biology, bioorthogonal transformations, and look at doing labeling studies. And potentially we can use these changes in emission property to maybe also have additional information about what's happening and where it's happening if we go into more complex systems. So I didn't do any of the work myself, so it just, in the lab at least, so it just leaves me to acknowledge the people that did do the work. So this is my current group, but the people whose work I've spoken to mostly through today are Eva, Richard, and Graham. Eva and Richard did the work on the bipyridylalanine, with Habis helping with some of the computational work on that system, and then Richard and Graham worked on the triazine system. I've mentioned a couple of collaborators, for example, Julia for X-ray crystallography, mass spec has been super helpful in this, and of course for the triazine work, Valerie and Andre at the University of Northumbria for kind of setting us off on this new direction and just asking the question, could we do this? Which hopefully we've shown that yes, we can, and now thinking about where we take it further. So thank you for listening and happy to answer any questions.
Great. Thank you, Amanda. Really fantastic talk. Very exciting science. We have a few minutes and happy to open any questions to the audience. I suppose I can ask the first question, but maybe I missed it. This triazine amino acid that you're encoding, will it also bind copper in the same way that the bipyridyl will? And do you expect it to be able to carry out that sort of Friedel-Crafts alkylation either before or after reaction with TCO? Is that a hope or not? Or am I misunderstanding the chemistry?
So I haven't ever thought about combining the two systems for catalysis. I mean, you certainly could. I guess the triazine system is just a different type of metal ligand. So we haven't looked. So we have thought about kind of first row transition metals and what effect they would have, particularly because you might think that if they were also able to speed up the bioconjugation or the click reaction, that's going to be more useful in a sort of cellular in vivo context. I guess the slight thing that I kind of hid there is the amino acid is not the easiest to synthesize, and so we've done all this work with kind of less than 100 megs of amino acid, and so that's kind of one of the roadblocks to going forward with this work. But yes, in theory, you could start to look at it as a ligand in its own right and not worry about it doing any click reactions, or using that handle to then change the properties of the metal complexes in other ways that you might want, whether it was for catalysis, whether it's photo properties and things like that.
Yeah, very cool. We have one question from the audience, Emerson Carmona. He says, it is a nice work, thanks. I was wondering how bright are the iridium complexes? What are the extinction coefficients and quantum yields of the complexes?
Which would be great if I remembered. So off the top of my head, I do not remember what the extinction coefficients are. The quantum yields of some of the iridium complexes, not with the triazine, but with the bipyridines, do change in the protein complexes and seem to increase versus the complexes themselves. And I think we've always been expecting that the extinction coefficients would track with the small molecules. But I don't know that we've shown that that's definitely the case.
Right. Great.